Hello, in this video we will discuss about the real-time PCR. So the PCR means the polymerase chain reaction, which that is done by the thermocycler, which that is a machine. So the PCR is used for the amplification of DNA segment, which that can be a small segment can be amplified. So what is the meaning of real-time PCR? So that is the same PCR, but it is modified PCR, which that is PCR converted into the real-time PCR. So here is the thermocycler, which that is basically we will discuss first our view of the real PCR just to understand. So here is the graph which that is showing in the thermocycler, which that is showing in the DNA denaturation during the cycle time and going to the cycle time, and the temperature when will be near to the 100 degrees Celsius. So the DNA will be denatured. After this denaturation, the temperature will be slightly down with automatically PCR change, and that's primer annealing, primer bind to become a annealing formation. So that means the DNA formation. And the primer extension is the next one, which that is slightly increase the 72 degrees Celsius temperature to increase the extension from the primer. So the primer means that is the specific primer which that is used for the DNA amplification. So here is the DNA, which that is two segment of DNA which that is amplified with the one cycle to multiply copies of the same DNA, which that is the equal to parental DNA, which that is given by the sample. So next, PCR plus spectrofluorometer or spectrophotometer is used for the with the PCR by using a probe or something. So that is known as the real-time PCR because it is show the color with the every cycle to increase color or decrease color depend on the experiment. So here is how does it work. So let's begin to understand how does it work. So here is the PCR machine that is known as the thermocycler. Here is the part, different part. Here is the part for the sample putting, and here is the part where the light will be fall on the spectrofluorometer. So this spectrofluorometer is move, accept the light and move toward where the DNA sample. So every cycle that light will be increased. So let's begin to, here is the mechanism of the spectrofluorometer in real-time PCR. So let's begin and we will also understand. So here is the monochromator are present, which that is light is striking. So here is a different color of light will be produced, but I am just understanding you. The light is made up of 7 color, but here I am not drawing that things, but here is the light extraction. So this light extraction is basically one part of light which that is different color, which that is desirable. So that means here is the basically a minor groove which that is attached the spectrofluorophore. So this spectrofluorophore is different. We will also discuss about that, but first of all we will discuss about the mechanism. So here is the excitation of the one light, and that is the monochromator is basically allowed to move one beam of light. So that is, here is the sample which that is present here, and that sample contain a color which that is our desire or our interest. So that's emit the color, emission of different color, for example green or blue. So this blue is a monochromator, another monochromator is put, and detector is used for detect with a computer or PC. So how does it work? We will discuss how does it work. So that is the question. So let's begin to understand. Basically here is the DNA parent DNA is it. So that is specific gene containing our interest, which that is we inoculated from different desirable gene where the defective gene are present, a cancerous gene. So that is, So here is we will take a EP tube, and that is here is the DNA polymerase is used for it, put into the tube or test tube which contain our DNA sample DNA, and detector we will add. What is the detector we will also discuss, and primer we will also discuss add in it, and DNA content which that is adenine, cytosine, guanine and thymine, and also add the detector. So what is detector? We will discuss. So the detector is in two type, non-specific detector and specific detector. Non-specific detector which that is detection fluorescence dye, which that is simple, and cyber green is a major important non-specific detector. Cyber green is produce as green color, but it is not produce another color, so we can check the amplification by the exponential increasing of the green color. While the specific detection detective probe which that is used, it is also known as TACman. This TACman contain a different type of color which that is our own interest with the related to our gene of interest. For example, different probe TACman contain a different specific TACman, so for use for the annealing process. So let's begin to understand the cyber green first. So the cyber green is the non-specific detector which that is produce the green color. So here is the DNA first parental, which that is at the minor groove site to attach the basically minor groove site to attach this cyber green. So cyber green molecule is more and more are present, but it will be bind when amplification will be more. It means the DNA content will be more, so the specific minor groove site will be increased, so the binding will be increased and color production will be increased by the each cycle. So amplification of this color with the amplification of DNA segments. For example, the polymerase chain reaction is used for it. So it is advantages and disadvantage. Advantage means the easy to handle, sensitive and basically cheap, effective. But disadvantages also non-specific, cannot check different types of DNA in the sample. Why? Because it is non-specific and we can't check the DNA different segments. DNA can be other segments, so that means it is difficult. So that is why we are using the specific detection in the same cycle for the specific DNA. If that color will be not produced with the specific our interest, so it means we can say that this segment is not present in the thermocycler, so we should change. So that means primer is the specific primer means the specific detector. Specific primer is used for the TACman. This basically a probe contain a fluorophore as well as quencher. This quencher is attached with the primer, also fluorophore. When the quencher will be attached, inhibitor of fluorophore, quencher is the inhibitor of fluorophore, still the at time when the attached with the fluorophore quencher, we cannot see the color before binding the primer with RNA for reverse transcription. So quencher will be detached from the fluorophore, then fluorophore will be produce color. So it is easy. So the specific detection we will discuss in this method. So first of all, here we are understanding the polymerase chain reaction to understand with primer specific and produce color, how it's produce color. Let's begin to understand. So the denaturation step we will discuss 94 to 98 degrees Celsius temperature used to denaturation of DNA. This denaturation of DNA, that is the specific probe will be attached, which that is bind with a quencher as well as fluorophore. So here is the specific site of the DNA, which that is our interest. Bind TACman bind with the 3' to 5' direction, while the 5' to 3' is the template DNA. So because complementary, recognize the sequence because complementary. So this is a complementary and it is our interest. So we are detecting the specific segment of DNA. So let's begin to understand that is DNA polymerase is used for the polymerization and attachment of the nucleotide which that is put in our sample during thermocycling. So here is the tagman. So this tagman will be bind. So the second step is the annealing process which that is used for the DNA polymerase. You are already know about that. So you already know about that. Basically 75 degrees Celsius to 80 degrees Celsius is used for the DNA polymerization to annealing process and extension. So annealing to extension, basically extension of DNA. So the primer will be bind. So a primer is the basically specific when primer is cancer containing, cancer will be separate and the nucleotide is also of the probe will be separate. So what that means, the fluorophore will be separate. So the fluorophore when separate to produce color because it will be detached from the cancer. So that means when more and more fluorophore will be detached during the each cycle, it means the color will be produced and amplification of color will detect our own DNA interest which that is doing the chain reaction. So that means the fluorophore is more. So the fluorophore when more, it means our gene of interest will is amplifying more and efficient. So we can check that DNA containing gene containing DNA is our desired and that we can confirm. So the tagman is advantages of tagman is specific but multiplex real-time PCR which that is contain basically different type of color blue, red and yellow and we can identify easily that gene of interest. So several gene detection we can check in the same cycle. And disadvantages also, disadvantages is that hard to sequence because different type of color which that is difficult to sequence, but we can take a 2 or 3 probe with containing a gene of interest, three types, so we can check easily and it is expensive and probability of false result can be. So next here is important things that reverse transcription of the HIV virus which that we can also detect the RNA of HIV virus which that is basically we can check the infection of virus basically. So here is we take the messenger RNA from the virus and reverse transcription. So here is the primer will be add in the sample and RNA polymerase and DNA content will be add as well as reverse transcriptase enzyme is also add. So after this adding to go run the PCR. After the running of the PCR, complementary DNA will be formed. This complementary DNA when this basically RNase enzyme is used for it. RNase enzyme is used for it to break down the parental basically parental RNA. So when denature the parental RNA because it is specific, so that means the PCR complementary DNA will be formed due to the PCR. So real-time PCR this is, and this will be show also color and we can detect that color. After the detection of that color, it is we can check that infection and use for many reason. So it is able to use many reason, checking the cancerous cell, checking the virus infection as well as checking the different genes are present in the different part of our body to check the RNA when extract the RNA and we can say that gene is active. If RNA is not our interest, so it means gene is inactive, so we can detect. So here is the computer which that is used for the quantitative real-time PCR and which that is show the graph with the color. So this graph here is the vertical shows that the fluorophore quantity or fluorophore potential, while the horizontal shows that the cyber number. So the number of cyber basically increased to increase the fluorophore, increase the color. So here the CQ value is here, and CQ value we will discuss, and also threshold line I am drawing. So the threshold line is very important. When when cross the threshold line, it means our gene of interest is going good and we are getting the positive result. And here is the exponential phase, which that is and indicate that our amplification of interested gene will be amplifying efficiently, and non-exponential curve will be occur. It means the our interest will be done. So thanks.